So welcome everybody. My name is Louise Labattoir from the Freshwater Biological Association, and welcome to this next instalment of the FBA seminar series. I'd like to formally welcome Professor Steve Ormerod. Steve is currently Professor of Ecology at Cardiff University and is Deputy Chair of Natural Resources Wales, a member of JNCC, Vice President of the RSPB and Chairman of Buglife. Steve's going to talk to us today about his career path to date, including his work on the effects on freshwater systems of acid rain, changing land use and global warming. So thanks again, Steve, and take it away when you're ready.
Thank you, Louise, for that introduction. I'm speaking to you from a very wet Cardiff this morning, and what I'm intending to do is talk a little bit about my entry route into freshwater sciences from some very early beginnings, and I'm going to be recycling some very, very old material. So if there are any students or early career people watching, this is not how you do a presentation. And I am going to come on to update some of the longer term work that we've been doing around acidification, climate change, and I'll look at some new stresses that we're starting to look at, in particular in the urban river environment. So the broad theme is around global change and river ecosystems, exemplified by some particular cases. This is where it began for me. I was born in 1958 in this terrace house in Burnley in Lancashire. This was my bedroom, but I was very fortunate. I had an elder brother who owned, he was 9 years older than me, and he owned a copy of the I Spy book of birds. And it was my brother David who essentially taught me how to identify the difference between male and female house sparrows that came and sat on our wall, and probably that was the earliest piece of taxonomy that I was able to do. My father was actually a mechanical engineer. There was some mechanical engineering at this stage in Burnley, but it just so happened that he had an apprentice, a man called David Crossley, who was, this is David probably about 5 years ago, a very keen naturalist, photographer, and amateur filmmaker. And David and my dad essentially were responsible for taking me out of this very industrial town, Burnley, and showing me something of the world beyond it. So our rivers in Burnley, this is the River Brun, passing through the town centre from which Burnley gets its name. Burnley was a major producer of cotton cloth. Cotton mills were very widespread. Our rivers were grossly polluted, very seriously modified, but not far beyond the confines of the town in the Thurston Valley, going on to the borders of Yorkshire, beyond a little town called Harle, Syke, was the River Don. And it was in this place that David and my dad would take me out fishing. There were some nefarious activities involving tickling trout under some of these overhung banks. And I remember one particular evening going out and with our hands catching 15 brown trout, some of which were returned and a small number were taken home to the table. This is now probably over 55 years ago. David and my father also took me out to the River Ribble, and Stainforth Foss, just outside Settle, was famous for its run of Atlantic salmon. These two images at the top and to the right are captures from videos made more recently by David Crossley. But probably at the age of 6 or 7, this was to me a major introduction into the character of rivers. And it was at that same place, Stainforth Foss, where or Stainforth Foss, to give it its proper name, I first saw the dipper. So at this stage, I could identify house sparrows, swallows, and dippers were probably my third species of bird that I could identify. And of course, much more interest in dippers follows down the line. Well, time passed, things went on. I went to be a student at Huddersfield Polytechnic, as it was then, but in an external professional training year, went to work on the North Downs in Surrey with the Field Studies Council at Juniper Hall, famous for fantastic arrays of orchids on local grasslands. But also I was given the job of cataloguing writing a key to the freshwater invertebrates that occupied all of the different teaching habitats that Juniper Hall actually used. And I was, armed with things like Kitten McCann's Guide to Freshwater Invertebrate Animals, and he and I exchanged some correspondence at this particular stage. But this now I'm a kind of, in my early 20s, 21, and I'm starting to get a bit of an interest in freshwater invertebrates, freshwater environments. I went back to Huddersfield to carry out my final year project. Huddersfield had a connection to Manchester via the Huddersfield Narrow Canal. And at this particular stage, the canal has now been restored, as the images on the right-hand side here show. But back then in the mid-1970s, all of the former locks on the canal had been what we call cascaded, and they represented this very marked local switch between fast-flowing conditions, lotic conditions, and lentic conditions downstream, associated with things like vegetation development. There were also very substantial changes in oxygen dynamics. And my project involved looking at the transition from lentic to lotic and back to lentic invertebrate assemblages. Now, at this stage, we didn't yet have the FBA keys to CADIS, but we did have some early taxonomic information. People like Peter Harle and Ian Wallace were already working on the identification of case CADIS. John Eddington, of course, who supervised Alan Hildrew before they wrote the key together on caseless CADIS, were already coming up with some kind of taxonomic information that I put to use in working out what the communities of organisms in the canal actually looked like. Well, time went on a little bit further, and I came down to Cardiff. At this stage, we had something like 70% of the rivers in the South Wales coalfield grossly polluted by a combination of poorly performing wastewater treatment works, trunk sewers that followed the river valleys, because the valleys are steep V valleys, but the trunk sewers very often leaked raw sewage directly into the river. We had the issue, of course, of active collieries at that stage, many of which gave rise to problems with fine sediments and iron ochre on the beds of rivers. And in Cardiff, however, we also had this man, Ron Edwards, who probably a lot of people will know and remember, who was instrumental in setting up a very active group of people interested in rivers, in freshwater pollution. And it was because we had such a problem with river pollution in South Wales that an MSc in Applied Hydrobiology was initiated. And I came to be a student on that course 40 years ago, in fact, in September. So I've been in Cardiff now 40 years. This is a colleague of mine, Ian Boothroyd, who was on the course with me, who went on to work on chironomids in New Zealand. And this is me in my early 20s back in those days. Well, of course, one thing leads to another, and from an MSc where I carried out a project on the Wye River system, I was given the opportunity to do a PhD by Ron Edwards. At this stage, we were discussing the possibility of the enlargement of Craigock Reservoir up on the Upper Elan in the top part of the Wye catchment. The notion was of enlarging Craigock, increasing the storage of water, increasing the compensation flow down the Wye. The water would be taken from the Wye into the Severn, out of the Severn into the Thames, and sent downriver to the southeast into London. Now, of course, that would have led to very substantial changes in water quality. And I was essentially, for my PhD, given the task of trying to work out what those changes in water quality might mean for aquatic invertebrates and other organisms. The Craigock scheme is still somewhere simmering in the background as a possible water trading, water transfer means of increasing the water supply into southeast England. This hasn't actually gone away completely, although of course it would be extremely controversial if it were ever to go ahead. Some of the early papers that I started to write with Ron, essentially started to evaluate what relationships between communities of organisms, of invertebrates in rivers look like in relation to water quality. We were at this stage using the same methodologies that were coming out of the FBA River Communities Project. And we showed, for example, this is just a very early figure from a paper in 1987, looking at how changes in water quality actually mapped on to this classification of invertebrate communities, which were created by the technologies of the day, twin span and Decorana. Now, what other things also happened at this time, and at the same time as I was looking at freshwater invertebrates in the Wye, the RSPB were actually counting dippers and grey wagtails and gooseanders and other river birds along the same rivers. And just by a series of just amazing coincidences, we were able to put the different data sets together and illustrate how birds like dippers were much more abundant where their favoured prey were abundant. These were things like caddis and mayflies. To understand that further, we also had to understand exactly what prey the birds were using. And my supervisor, Ron Edwards, was driven absolutely apoplectic by the fact that the papers from my PhD and my PhD was delayed because pretty much as an amateur at this stage, I started to write papers about these fantastic river birds. And, you know, this is almost a parallel career to my work with freshwater invertebrates. But we've illustrated, for example, just what a potential top-down effect predation by dippers can mean for the abundances and the production of invertebrates in the river environment. And I've been incredibly fortunate through my career to travel around the world looking at 4 so far of the 5 dipper species, but also working on bird communities in the Himalayan mountains, bird like these white-capped river chats and forktails and plumbeous redstarts that are all really integral bird communities associated with the river environment. And I won't really be talking very much about that today. I may touch on it later on. Well, as I started to travel around the world, going to conferences and looking at other river environments in the kind of late 1980s, I had a Winston Churchill Fellowship. Of course, and also partly through Ron Edwards, Ron was very interested in catchment scale effects on rivers. At a very early conference at the BES in Leicester in 1984, I was introduced by Jean Likens. I knew the very famous work by Noel Hines on the stream and its valley. And you know, the idea was abroad, of course, that the character of river catchments, be it their geology, their continental position, the vegetation that they had, the soil types, the amount of rainfall they receive, the temperature, would all have a substantial influence on the rivers that drain from those same catchment areas. And this was an idea that marked me very, very substantially, but it also started to strike me that because the characteristics of river catchments were so fundamental in influencing downstream character in rivers, if people therefore started to change the attributes of catchments, there was the potential for very substantial, large-scale influences on river environments downstream. And between 1989 and 1999, I worked on and off in the Himalayan mountains. This is an image that I took flying over the middle hills of Nepal. We had a study site in the Liku Kola, which is the river crisscrossing this particular image. We also work regionally across the Himalayan mountains from east to west. But I think what you can see in this image is how significantly the natural vegetation of tree cover has been replaced by agriculture, and you can see pretty extensive terraced agriculture right the way across this image. that had significant effects on the use of agrochemicals, on erosion, on water retention. And so there were just some kind of representations of the kind of anthropogenic effects that catchment modifications by people might have downstream. And we know in these upper reaches of the Ganges now just how substantially and with what cumulative effect those modifications to catchment character have actually looked like. So I guess one of the things I wanted to do at this stage was to find a way of investigating just what kind of effects modifications at whole catchment scales might actually look like and might start to lead to downstream. But of course, the spatial scales and the temporal scales involved in these changes were so dramatically large and long-term that to think of doing this using experimental methods was really, really challenging. So ideas like space for time substitution became quite important in the armory of techniques that people used looking at these kinds of problems. And here's a kind of a very early cartoon that I put together illustrating putative variations in catchment character in what might be upland Wales. It could easily be somewhere else. But it was possible to select certain sampling points across this array of different catchment attributes, at least in low order streams, to come up with contrast where you could compare catchments that had relatively consistent character. And somewhere within this hierarchy of locations, you could choose sampling points for biology or chemistry to give you the kind of contrast that you wish to see. Now, of course, it's also possible that these different subcatchment units could also be manipulated. I'm going to come on and present an example of that in a few minutes' time. Now, because this was essentially a correlative approach, this was about space for time substitution, there had to be process studies that somehow would reinforce and add additional evidence to things that were revealed by correlative work. And so, for example, hill slope hydrology or hydrochemical investigations in soil water or examinations of exchange of material such as allocthenous carbon, the movement of organisms across river basins were all additional things that you could use, that the investigation of processes that somehow could support the weaker, more inferential material that came out of the comparative approach. Of course, also there was the potential, because these were large-scale, long-term effects of assessing how patterns in different catchment characters actually evolved through time. And of course, any kind of problem at this scale, large scale, long-term changes in land use or climate change or acid deposition or pollution are always benefit from a perspective that involves collecting data across years. My models in all of this were still working through food webs up to things like birds, but very substantially aquatic invertebrates, heptogenic mayflies. Here's a net spun by a hydrosyke caddis that I photographed in the Afon Methlin in upland Wales, river limpets and silus. So it was the invertebrate community that interested me largely, but I did work also on diatoms, occasionally on fish, and wherever we could on the kind of process interactions between these different groups of organisms. So I guess the first major global change problem that I had the opportunity to look at was starting in the late 1980s. At that stage, we were deeply interested in the effects of acid rain on the freshwater environment in both base pore areas like Wales. These data were put together in the late 1980s. They were first presented at a conference at what was then the Institute of Terrestrial Ecology on the effects of conifer forestry on river environment. But out of our Welsh acid water survey that had been carried out in 1984, we were aware that metals such as aluminium were toxic to invertebrates. We were also aware that concentrations of aluminium increased in base pore locations where conifers had a large cover on the catchment, and the richness of organisms of ephemeropterans, trichopterans, and indeed all invertebrates across these upland Welsh sites declined as aluminium concentrations went up and pH went down. And really, it was out of this apparent influence of acid deposition that the Llynbrioni experiment, the Llynbrioni catchment project, was born. What we now call the Llynbrioni Stream Observatory. So at this, in the late 1960s, 1969, Llynbrioni Dam was completed as a means of regulating the River Tawe to supply water for Swansea. The Tawe was a major river for sea trout and extremely important large sea trout catchers, but concerns about the lack of access to upstream spawning sites led to an idea that as fish were migrating upstream, they could be caught in a trap, put into lorries, transported around the dam, released upstream, whereas the smolt coming downstream again could be trapped and trucked around the reservoir and released downstream. So what was called the trapping and trucking scheme. That scheme through the 1970s very palpably had been failing, and even though fry were also being planted out as well as fish occasionally released upstream, no smolts were ever caught in the downstream traps. And that led to an investigation of what it was, what attributes of catchment upstream of Llynbrioni Dam might somehow be involved in this clear failure in this fisheries management scheme. So what was put into place by John Stoner, Alan G, Ron Edwards, who at that stage were all part of the Welsh Water Scientific Directorate, was the Llynbrioni project, where catchments were selected either in conifer forestry that were acid, upland, moorland that were either circumneutral or acid, and we had a small number of deciduous woodland catchments. And I was given in 1984 the job of running the group of people, postdoctoral people, who would look at the chemical and the biological ramifications of these variations in catchment character. So the observatory was born in 1981. Some of the catchments were manipulated, and I'll talk a little bit about that in a minute, but others have been reference catchments, and nothing has been done to those catchments since 1980. And because of underlying geology, there is a variation from streams that are acid with low numbers of invertebrates through to those that are circumneutral that are richest in species. What the data we collected at Llynbrioni allowed us to start to develop were models through which we could relate the chemistry of the different stream environments, both to the richness and the community composition of organisms. So again, using early multivariate methods, twin span, Decorana, we could map out what communities of organisms look like in relation to chemistry. We could work out what numbers of taxa occurred in streams with different attributes. And across the Welsh region, we could get a handle on the tolerances to pH that different invertebrates appeared to have. Now, here's the kind of step to manipulative investigations of catchment character. So one of the things that Brioni was set up to investigate was partly the influence of land use on acidification, but also whether there was any symptomatic treatment that could be used to reduce the acidification problem. And the models that we had actually offered an opportunity, because we knew what the communities looked like in different conditions, what richnesses looked like. we were able to make predictions that to get recovery from acidification, there had to be an increase in pH, either as deposition declined. There also had to be a gain in species, but the other option that we had here was to try and accelerate acidification by liming some of the catchments around Brioni Reservoir. And indeed, those additions of calcium carbonate went ahead on 3 different catchments. And what this figure is illustrating here is what has happened to calcium concentrations in our manipulated catchments by comparison with adjacent references. And very clearly, there is a very substantial response, with calcium concentrations being sometimes, in these cases, doubled or more. A persistence through time and in at least some of the catchments, a rather more short-term response in others. But of course, now the major question is, to what extent has there been a biological response? And again, these are data we published back in 2009. What we're looking at here in the top part of the figure is a representation of the community space occupied by invertebrate assemblages in relation to chemistry. And at the bottom of the figure, we're looking from 1980 through to the present day, going forward up to 2005 to get a handle on whether the assemblage compositions have changed in our different stream types. And what we could see up to 2005, when we last looked at these data using these methods, acid forest streams really hadn't changed very much. Circumneutral streams, of course, our major references, hadn't changed very much. And even in streams that were deacidified by liming, or were recovering because acid deposition was abating, the influence on invertebrate assemblages by this stage was actually very small. We probably gained about 3 or 4 acid-sensitive organisms. So something clearly was creating a barrier to a more full and complete recovery. So I can actually now bring some updates to this work. I currently have a student who is actually looking, trying to update both the chemical record from Llynbrioni and the biological record. Natural Resources Wales still sample the streams around Llynbrioni. And what we're looking at here are trends in mean pH, in this case, in acid forest streams, in acid grassland, and in circumneutral streams. Through time, these are unmanipulated reference catchments, and superimposed on these figures are also 3 shaded grey bars that represent the mean values for the same stream types across the whole of Wales from our acid water survey. About 70 different streams are involved in the means for the grey bar. So what you see here is that there is evidence of chemical recovery. So the pHs have been progressively increasing, consistent with reductions in acid deposition through time. But equally, you see here also that round about 2014, some kind of reset event occurred. And back when we published some work in Journal of Applied Ecology in 2009, we had already illustrated how very wet conditions, wet winters, were enough to eliminate substantial parts of ongoing recovery from acid rain effects. And indeed, 2014 was up to 2014 the wettest winter. 2013-14 winter was the wettest on record up to that point. And these are just 2 sets of Met Office data, one indicating rainfall pattern in February 2014, and the right-hand figure indicating the percentage change in 2014 over long-term averages, and the darker blues indicate the bigger variation from long-term average. So we're still dealing with circumstances where, at least in pH terms, there can be some kind of reset associated with climatic conditions. We're investigating further what this event looked like for other key aspects of stream chemistry, such as aluminium concentrations, and we also, of course, have data on things like the effects on minimum pH. So what about the biological story? Well, Again, Alex Carr, my student, has been looking across Wales using the Welsh Acid Water Survey data, as well as looking at Llynbrioni. And at the moment, we've got as far as using the Acid Water Indicator Community Score. We're going beyond this to use other methods. And what you see here is that in acid forest, acid moorland, circumneutral streams, there is some indication of increase through time, at least in acid forest streams, and actually also in circumneutral streams. So the values in 2012, when we recorded them, were higher than in the previous two surveys. At Llynbrioni also, there is evidence of increasing positive regression relationships between the A-week score and the trends through time. However, these are quite modest and still quite small changes. And again, at the moment, we're unpicking the community response to assess just exactly which organisms are contributing to this trend and what they tell us about the response from acidification. Now, Alan Hildrew and I have been talking about different potential explanations for what appears to be a partial recovery from acidification through time. And Alan, if you're listening in, I think we're moving closer to be able to pick up the text that you sent me about 3 years ago. But there are different potential hypotheses around here. Is it that habitat's appropriate? Is there a biological exclusion for some reason? Dispersal and recolonization might be involved. Are we seeing continuing episodes? Are we overlooking some stresses? Or is there a potential climate change effect that is now subsuming the recovery patterns from acidification? So climate among those is probably the biggest issue that we've looked at Llynbrioni, partly as a means of understanding recovery from acidification, but also as an issue in its own right. And the Brioni project, now being there for 40 years, gives us a means of getting some kind of handle on what long-term trends at Llynbrioni look like and what they lead to in biological terms. This is not a straightforward issue. There are increases in temperature in the Brioni streams that are consistent with global warming, but there are also patterns that track the North Atlantic Oscillation. So here's some data that we put together back in 2007, looking at how stream temperatures at Brioni track the winter North Atlantic Oscillation Index, this pattern that tells us whether we have warm, wet winters or cold, dry winters. And the first real hints we had that these big climatic systems influenced organisms in our streams came in 2001, where Dave Bradley looked at the persistence of invertebrate communities, the year-to-year stability in relation to the North Atlantic Oscillation Index. So what we're looking at on this figure is Dave's representation of, first of all, the NAO in these grey symbols. And in the late 1980s and early 90s, there was this big positive amplification of the NAO that brought wet, warm, stormy conditions, interestingly enough, followed by droughts in the following spring and summer. And what we're actually looking at in these black triangles is the year-to-year similarity of invertebrate assemblages tracking the North Atlantic Oscillation. And that pattern was apparent in all of the stream types at Llynbrioni. So some cross-catchment influence had led to what appeared to be a loss of interannual stability, where we had this sequence of warm, wet winter periods. Now, however, something else also came into focus, and Isabel Durance and I in 2007 brought together data looking at the relationship between invertebrate abundance, temperature variations partly caused by trends through time, partly caused by the NAO, and also abundance and patterns between 1980 and 2005. And what Isabel's analysis showed, at least in our circumneutral streams at Flindriani, was that it appeared that abundances of invertebrates fell by about 20% for every 1 degree centigrade rise in stream temperature. Now, of course, there were other things potentially involved in this, increased discharge under some circumstances. It was possible that this could be a phenological effect. It could be a top-down effect by fish whose energy requirements were larger at higher temperature. But we also became quite interested in the effects of litter dynamics and litter potentially being exported more rapidly at high flow, but also breaking down more rapidly at higher temperature. And I'm going to come on to that in a minute. But the next phase again is just a bit of an update. Fiona Joyce, one of my PhD students, is currently updating the trends at Flindriani. The reductions in invertebrate abundance appeared to have continued through time. They appeared to be apparent in all of our different stream types: acid moorland, conifer, circumneutral moorland, broadleaf. Fiona is looking at the kinds of land use effects that actually prevent or at least restrict the extent to which invertebrate abundances are declining. So her PhD very much is about stability and resilience patterns. Nevertheless, these longer term trends in invertebrate abundances appear to be at least continuing. So something is still stressing the Flindriani streams. Just to go back to the issue about land use and litter dynamics, these are some figures taken from Marion Pye's PhD thesis. And Marion, over a 4-year period, looked explicitly, and this is the 4-year period of varying rainfall pattern. She looked explicitly at the inputs of litter into the Brioni streams, what the exports look like under different flow conditions, and also what the storage looked like in our different broadleaf, conifer, acid moorland and circumneutral moorland stream types. We're still actually bringing this analysis to conclusion for publication, but the broad inference at this stage is that litter inputs, of course, from broadleaves are bigger than anywhere else. The standing stock of litter in the broadleaf streams is bigger than anywhere else, despite export, which is large at high flow. And that has taken us to the view that it is possible that the management of riparian zones by the maintenance or re-establishment of deciduous woodland could act as a means of offsetting some of these potentially negative effects on litter stocks brought about by warmer, wetter conditions. And these are some figures from Steve Thomas's PhD. Steve looked in the Brecon Beacons, in fact, at streams with different catchment character, brown trout abundance in streams with riparian buffers, conifer, deciduous woodland or moorland, and we think that there were other influences on brown trout abundance. Conifer was the worst of all options. But when we looked at invertebrate biomass, very clearly either in deciduous woodland or streams with deciduous buffers, there appeared to be a way that the standing stock of invertebrates was somehow enhanced or maintained where we had these riparian broadleaf trees in that riparian zone. Now, of course, there are other things than climate change going on in the upland river environment in Britain. And this gives me a bit of an opportunity just to talk a little bit about birds, because the data that the British Trust for Ornithology bring together through various monitoring schemes and through various atlases give us an indication of not just the extent of change, but the geographical pattern of change in the occupancy of monitored locations by river birds like the dipper and grey wagtail. Anywhere that's blue on these maps are areas where the populations, the distribution has thinned out. anywhere that's red is where there has been an increase in occurrence. And there is really quite an interesting pattern here, because you see down, for example, in the South Wales valleys, an increase in the occurrence of dippers. And we now think that is associated with the recovery of urban streams from past problems of organic pollution. But more widely, in the rural river environment, there appears to have been a thinning out of dipper populations. And that's shown quite clearly in this long-term index, where the kind of overall population appears to have gone down by something like 20 to 30%. That decline is even bigger in grey wagtails. And again, I now have a PhD student who is looking in more detail at what the land use drivers of these trends actually might be. But again, we have looked a little bit at the invertebrate signal that is coming from different elements of land use pattern in the farmed environment in Wales. And Stefano Lawson in particular, another PhD student of mine, just looked at one particular dimension of this, and that was around sedimentation associated with bank face erosion and cattle poaching and various other sources of sediment. Stefano's data illustrated, for example, how richnesses of invertebrates declined as as sediment cover increased on the bed of rivers. So mayflies, stoneflies, caddis declining with increasing suspendable sediment, but potentially even more significantly, the losses of different invertebrates appeared to be associated with the particular traits that organisms had. So organisms that were filter feeders or required a clean surface from which to graze algae appeared to do less well in streams where sediments were an issue. But also he showed that where there was more intensive catchment land use for agriculture, we also appeared to be systematically losing larger, long-lived organisms that required stable conditions in which to complete their life cycle. So a systematic dropout from streams in which catchments were being converted to agriculture, or where sediments were a problem. And I would maintain that if we saw some kind of systematic conservation influence like that in the terrestrial environment, certainly it would concern us because it would be more conspicuous. But of course, things beneath the water surface very often are out of sight and out of mind. Other issues in the Wye catchment and parts of rural Wales have really been reaching the news recently, and I don't know how many people have been following George Monbiot's items, for example, on the River Wye and elsewhere, where there is now this apparent issue with algal blooms, which the Rivers Trust, Wyanus Foundation and Afonydd Cymru are drawing our attention to. There are issues with other kinds of benthic algae potentially replacing the ranunculus, for which the River Wye was notified as a special area of conservation. And a lot of the finger at the moment is being pointed at intensive poultry units. And on the borders of Herefordshire, Radnorshire, the borders of Wales, the density of these intensive poultry units is larger than anywhere else in the United Kingdom. And we are at the moment looking quite closely in NRW around just what we may be able to do around planning constraints or planning mechanisms to somehow regulate what could be having adverse effects. I think we need to understand some of the biological consequences further, but again, just to remind you of some very, very old data going back into the late 1970s and early 1980s. People like Tony Jenkins were producing these fantastic invertebrate inventories of places like the Teivi. Mike Brooker and Dave Morris did exactly the same in the Wye system. And I think we really have a major opportunity to go back and compare how the Wye and the Teivi and these other fantastic Welsh rivers now look like by comparison with their past conditions. So turning just to start to bring things towards a close to some work we've been doing in the urban river environment, Ian Vaughan and I, now probably about 8 or 9 years ago, looked at agency data from England and from Wales, these huge holdings, 200,000 samples, 20,000 locations, 20 years of data, to illustrate that at least in the urban environment, there appeared to be a recovery ongoing from the past influences of gross organic pollution. And what we're looking at on this figure is an indication of how richness, family richness of invertebrates across English and Welsh streams had progressed through time. Also how community composition had changed, here illustrated by ordination scores. The Environment Agency have just asked us to update this analysis, so we're hoping sometime next year to have a little bit more to say about this. There is some suspicion that the recovery may be stalling on the back of effects from things like combined sewer overflows. But I guess one other thing we ought to be aware of is that the problems of the urban river environment are by no means solved. And we still have issues with, for example, accumulating legacy contaminants that occur within food webs. These are some data from my most recently graduated PhD student, Fred Windsor, looking at how things like flame retardant chemicals, PBDEs, accumulate between primary producers, basal resources, right up to dippers. PCBs also accumulating in similar ways. And if you look, for example, at the concentrations of things like PBD flame retardants in the eggs of birds like the dipper, they are at their greatest in urban areas. And in fact, the concentrations of PBDs we record in dippers in the South Wales urban valleys are some of the highest that have ever been recorded in passerine birds, in songbirds, anywhere on Earth. And there's a bit of evidence that they have some kind of an adverse effect on chick development. Some of the most recent work we've been doing on new emerging contaminants is actually on the issue of plastic. So within the world's oceans, of course, plastic has now become a real cause célèbre on the back of David Attenborough's Blue Planet 2. But it's worth remembering that of all the plastic that is in the world's oceans, something like 8 to 12 million tons entering the world's oceans every year, at least 4 million tons is actually coming through rivers. And in the river environment, of course, it's much closer to its sources. So the concentrations per unit volume of water are liable to be much higher. And we think that something like 1 to 2.5 million tons of that plastic coming through rivers is in microplastic form, fibers, tire dust, road paint, and those kinds of materials. And these are data that came out of colleagues in Manchester, illustrating that in the sediments of rivers like the Irwell system, in fact, we find some of the highest microplastic densities ever measured in the benthos of any location anywhere in the world. And in this particular instance, it was about 500,000 microplastic fragments for every square meter of riverbed. And anyone who knows invertebrate densities in the benthos of rivers know that is a pretty large number. Well, we started to look back in 2016 at what some of this apparent concentration of plastic in the river environment might look like. We were interested in whether places such as wastewater treatment works or CSOs could be a source of plastic, whether it occurred in any way in invertebrates, and whether it might find its way through food webs into birds such as the dipper. Again, Fred led this work. It was initially carried out by somebody called Rosie Tilley, who was a professional training year student. And we looked upstream and downstream of wastewater treatment works in the Taff and the Usk in the Wye, in heptogenids, in bitids, and in hydrosychid caddis. And essentially what we found, I think, really staggered us, that we could find plastic fragments everywhere. It wasn't just downstream of sewage outfalls, although it was in greater concentrations where wastewater contributed more to river flow. And we find plastic in all of these 3 different invertebrates that, of course, are occupying different places in the base of river food webs. 2 grazers and 1 filter feeder, so plastic appears to be entering food webs in more than one way, but staggeringly, plastic was present in half of all the insects we sampled at this array of sites, mostly fibres. Of course, we then went on to ask what that might mean for ingestion by dippers, of course, which are dependent on exactly those same materials. This work was carried out by a guy called Joe de Souza, again supported by Fred and David Santillo in Exeter, who identified the material we found. This was wide-scale study in urban and rural bits of different rivers. And at 14 of the 15 sites, we found plastic both in the regurgitates and in the feces of dippers, mostly polyester fibres. And because we have really fantastic data on the energetics of dippers from work by David Bryant and others in the 1980s, we were able to calculate from the load of plastic in dipper prey just how much plastic dippers are ingesting, just how much they're also feeding to their chicks. And the calculations suggest that dippers are ingesting something like 200 microplastic fragments daily. They're feeding something like 5,000 to 8,000 microplastic fragments to their chicks before they fledge. At the moment, we don't know whether this has any effects or not. Could be transporting other contaminants, it could cause physical damage, could be toxic in its own right. But again, one piece of information that we were able to put together from energetics and from our assessments of the densities of plastic per unit dry mass of material was that the birds appear to be voiding plastics at pretty much the same rate that they're ingesting it. So as much is coming out as goes in the first instance, and this might be at the heart of this something of a good news story if this material is behaving in an inert way and if the birds aren't retaining any of this material. But we need to know much more about this issue. All of this work had really substantial political and news resonance in Wales. The National Assembly Climate Change, Environment and Rural Affairs Committee ran an inquiry on the back of the work we'd done, actually finding plastics in invertebrates and then in dippers. Fred and I led the evidence to that inquiry. So, of course, now we know we are entering a really serious era of biodiversity loss. And if anyone hasn't watched the David Attenborough programme on Netflix on current circumstances, I would really strongly recommend you do that. But the information coming out of things like the Living Planet Index from WWF illustrates that freshwaters across the world really are in serious trouble. We know that there are issues with the calculation of this index, but it is corroborated by other information. So this is work by Clement Tochner and Sonia Yarnig's group, illustrating, for example, how the global freshwater megafauna, sturgeon, and other species in freshwaters are absolutely plummeting. And the Chinese paddlefish on Earth for 200 million years and yet goes extinct in our lifetime. This is a tragedy unfurling before us, but hidden from us beneath the water surface. And with others around the world, we've started to look at emerging threats, what they look like, what it is we've got to tackle, but also we started to look at what the big issues are that we could use to start to get to grips with this tragedy of freshwater biodiversity loss. The notion of bending the curve that was given to the world by Georgina Mace not long before she died. But in the case of rivers, allowing rivers to flow more naturally, reducing pollution, protecting critical habitats, ending overfishing and unsustainable sand mining, controlling invasive species, safeguarding river connectivity. And I hope that all of us as a freshwater community can get behind this effort to disseminate the problem that we face and actually advocate some of the solutions that we can find to address it. So Louise, I did have a conclusion side, but in fact, I'm going to skip that and bring things to an end if I can now do that and invite any questions.
That was great, Steve. Thank you. Yeah, anybody, if you've got any questions, please put them in the chat box and we'll ask Steve those in turn. I had one to kick off, Steve. So going back to the work you were describing in the first kind of third of your presentation, do the effects that you saw on the invertebrates of the acid rain and the other effects in that subcatchment fully explain the failure of smolts to run back to the sea, or were there other factors that are kind of as yet unknown?
I mean, there was no reason why, in physical terms, upstream of Llynbrioni, the main stem of the Camddwr and the Tyve and the Tawe were in any way inappropriate. And in fact, in the Camddwr, there is a brown trout population that does hang on. So there's, there wasn't really any other reason. And acidification, toxicological information, fish exposure experiments, variations in density of brown trout across the streams very clearly showed that things like aluminium toxicity was really, really problematic in those streams. So brown trout that were planted out in ecological experiments lasted about 24 hours in those streams. They became heavily coated by mucus from the gills accumulated aluminium. Aluminium toxicity was the prime explanation.
Great, thank you. I've got a question from Mark Gessner. He says, official documents of the Forest Service in the 90s recommended sparse riparian vegetation along upland streams. The argument was that the tree canopies would intercept acid rain and thereby exacerbate the stream acidification, which he says puzzled him at the time. What's your take on how such fundamental misjudgment could happen at the time, and what's the situation now?
Yeah, yes, that was interesting, wasn't it? But those initial manipulations of forest cover, first of all, were dominantly associated with plantation conifer. The dominant wisdom at that time was that shading was a really serious issue for primary production within the streams. And I think actually there's some really bizarre evidence coming out of Ireland that illustrated that clearing woodland from stream banks increased in-stream primary production of vegetation. That was good for invertebrates, and the fish did better. But it appeared to be actually quite unusual circumstances. Even some of our own rivers trusts were actually advocating the clearance of trees from banks at that time. It also surprised me, Mark, at the time that people were advocating exactly that course of action because of what we understood from the river continuum concept and various other things. And I hope we've probably got back into a slightly better place. We understand that the benefits are multiple. They are around shading, they're around sediment retention, they are around denitrification, and very clearly, from what we show, they're also around allochthonous energy inputs.
Thank you, Steve. So another question is, for the UK as we enter the post-Brexit period, what are the 3 or 4 key measures that you would like to see incorporated into the agricultural payment systems in order to bend the curve of UK freshwater biodiversity loss?
Yeah, I saw that question pop up from Dave, and of course Dave Tickner has been instrumental in taking forward the bending the curve methodology into freshwaters, and I've worked with Dave on exactly that. Clearly, Dave, we've got to be radically modifying the way we balance catchment utilization, catchment exploitation, and downstream consequences. Some of that could be symptomatic around what we do in riparian zones, so at least you're trying to minimize the influence of the wider catchment on the stream. But I think also in what we know are critical catchments, sensitive catchments, we should be doing something to reduce the intensity of use patterns of all types. Now, of course, that is a really serious issue because we've got to balance food production with other elements of nature conservation. But if we really care about the freshwater environment, actually making sure that freshwaters are properly recognised in NELMs and in Glastear, our Welsh equivalent, I think is really critically important. I
think we have time for one more question. So this question is, Have macroinvertebrates adapted to anthropogenic influences in past centuries? And if so, would we still expect strong positive outcomes if rivers are restored to more natural conditions now?
What does adaptation mean in that sense? Is it a genetic term? If it's genetic adaptation, I mean, I'm not the right person to answer a question about genetics, but critically, there is a balance between the generation time of the organisms we're dealing with, and invertebrates can have life cycles that run over 1, 2, 3 years, sometimes less than 1 year. So the number of generations allowed for adaptation can be limited. Some of the stressors are large and rapid, and the acidification example was one which was large and very rapid and clearly would have required very substantial adaptation. If the question is more about a community adjustment, that modified streams are now occupied by different sets of invertebrates than might be there under what we call natural circumstances, the reinvasion of those communities could be a barrier to change and recovery. And this is one of the things that Alan, Hilda and I have been discussing through time. So we don't have the answer yet, I think is probably the answer to that question. So.
Thanks, Steve. I think that's all we've got time for. It's 1 o'clock. So thank you, everyone. And special thank you to Steve for a really interesting talk and interesting discussion there at the end. And yeah, round of applause from everyone there. That's great if you can find your reactions button. Thank you very much, Steve.
Okay,
and look out on the FBA's website for information about the recordings of these seminars, which will be up shortly, and for information on future FBA seminars too.